Yes, students, welcome to the present session. Here I will discuss about the structure of nucleotides. If we want to understand the overall structure and function of our genetic material, that is nucleic acids, we need to learn the basic structure of its building blocks, that is nucleotides. So, from the last session, we came to know that nucleic acids are biopolymers. And the basic building block of nucleic acids are known as nucleotides. Only two types of nucleic acids are present in living organisms. These are DNA and RNA. Now, in this session, we will focus on the structure of nucleotides. What is the basic chemical components of nucleotides? This is a simple diagrammatic representation of a single nucleotide. A single nucleotide is made up of three chemical groups. Phosphate, denoted here as violet circle in the left corner of this diagram. Next is sugar molecule. Next to the phosphate group, the phosphate group is actually covalently bonded with a sugar molecule. So here the sugar molecule is depicted as a pentagon, sky blue colored. Next one is the nitrogenous base, the green colored rectangle here. Now, one by one, we will understand the structure of each of these three components. That means phosphate, sugar, and nitrogenous bases. What are the different chemical variations are present in a nucleotide? First of all, we will discuss about the sugar. Sugar present inside the nucleic acid or single nucleotide is a 5-carbon sugar. Now, all of you are familiar with glucose. Glucose is nothing but a 6-carbon sugar. In chemical nomenclature, all these 6-carbon sugars are known as hexose. Hex comes from the number 6. Similarly, all the 5-carbon sugars are known as pentoses. Pentoses. The word pent indicates 5 carbon atoms in its structure. Now, so the pentose sugar, the core ring of the pentose sugar is made up of carbon and oxygen. So carbon and oxygen atoms. Now, how these 5 carbon are located in a pentose ring? The simple basic skeleton of pentose. Look at take a look to the basic simple skeleton of the pentose sugar. The blue pentagon here. And put carbon at each corner of this way, in this way. Carbon in this way. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Note that the last carbon, that carbon number 5, is actually located in outside of the ring structure. And here at the center topmost position of the ring, is actually contain one oxygen atom. So in this way, all the carbon, 5 carbon and 1 oxygen is located in the, combining and forming the ring of a pentose sugar. Now, what are the different types of pentose sugars are there? In our body, there are different types of pentose sugars, but 2 types of pentose sugars are present inside the nucleic acids. One is ribose, and the another one is deoxyribose. Now, what is the difference between these two pentose sugars? First of all, we have to add the functional hydroxyl group with each carbon atom in the ring. Then it will be converted into the functional sugars. It may be ribose or deoxyribose. Now you can now see the detailed structure of ribose and deoxyribose. You can see in the chemical structures that all the ring carbon atoms are denoted from 1 to 5 at a clockwise direction. So the carbon atom next to the oxygen atom is denoted here as carbon number 1, then carbon number 2, then carbon number 3, then carbon number 5. After that, the carbon number 4, carbon number 5 is located outside of the ring. And it is actually bonded to the ring carbon, that is carbon number 4. Now, next point is that all the ring carbon atoms are attached with one functional group, that is hydroxyl group. Now, see the difference between the structure of ribose and deoxyribose. You can see very clearly that in case of ribose, carbon number 2 is attached with the hydroxyl functional group. But in case of deoxyribose, this hydroxyl functional group at carbon number 2 is missing. Okay, so this is the very important difference between ribose and deoxyribose. So actually deoxyribose is containing or holding one bonded with one hydrogen atom instead of hydroxyl group. Now, the prefix deoxy hence indicates the removal of oxygen atom from the carbon number 2. This simple difference of one hydroxyl group make a significant change in its chemical function. Chemically, deoxyribose is more stable than ribose due to the absence of this hydroxyl group at carbon number 2 in the pentose sugar. Now, we have to know which sugar is present where deoxyribose is present in DNA. Okay, and the ribose sugar is present in RNA. To remember, you can correlate in this way that the starting D of DNA is coming from its sugar, that is deoxyribose. Okay, and in case of RNA, it starts with RNA and the sugar is here the ribosome. The next component is the phosphate group. In the nucleotides, you can see here the structure of one phosphate group. Okay, so one phosphorus atom is attached with 4 oxygen atoms. All these oxygens are actually negatively charged. Okay, so this phosphate group is highly polar group and negatively charged due to the presence of highly electronegative 4 oxygen atoms. Next, the third important chemical group present in a nucleotide is the nitrogenous base. Nitrogenous bases are basically a molecule composed of significant amount of nitrogen atoms. Due to the presence of nitrogen atom, it acts like a base. So, as a whole, it actually called as a nitrogenous base. All the living organisms are composed of basically 4 elements: carbon, oxygen, nitrogen, and hydrogen. In nitrogenous base, nitrogen atoms is combining with all these elements, forming a ring-like structure. So this is the basic skeleton here, represented in a diagrammatic way. So it is basic in nature as it contains nitrogen which can donate electron. So nitrogens are forming ring-like structure in combination with carbon. Now very importantly, it can form 2 types of ring-like structure. One, there is a single ring. Another type contains double ring. Okay, the single ring is called pyrimidines and the double ring is called the double ring structure is called as purines. Now, depending on the arrangement of the carbon and nitrogen in the ring, there are 2 different kinds of pyrimidines, 3 different kinds of pyrimidines. These are cytosines, thymine, and uracil. Whereas there are 2 different types of purines. These are adenine and guanine. 2 different. Now we will see the detailed chemical structure of all these 5 bases. Note one by one that all the pyrimidines have 1 ring and all the purines have 2 rings. All the rings of the bases are basically composed of carbon and nitrogens. Pyrimidine rings are composed of total 6 atoms, where purine rings are composed of total 9 atoms. Due to the double ring structure of the purines, these are the larger in size compared to pyrimidines. Now we have to see how these 3 individual chemical groups, that is phosphate, sugar, and the nitrogen base. Phosphate, sugar, and the nitrogen base bond to each other to form a single nucleotide. Now, look at the center molecule. Focus on the center molecule. That is the sugar molecule. Okay. So, the carbon one of the sugar is attached to the nitrogenous base. This type of bond, where carbohydrate or a sugar molecule is attached to another structure, is known as glycosidic bond. Now, in nucleotides, carbon one of the sugar is bonded with the nitrogenous base by one glycosidic bond. If the base is pyrimidine, then according to the chemical nomenclature, it is the nitrogen number 1, which is making glycosidic bond with the carbon number 1 of nitrogen number 1, which is making glycosidic bond with the carbon number 1 of sugar in nucleotides. And if it is purine, then it is nitrogen number 9, which is forming the glycosidic bond with the carbon number 1 of the sugar molecule in nucleotides. Next, move to the last component, that is the phosphate group. How the phosphate group is attached with the rest part of the nuclear, with the rest part. You can see that the phosphate group is attached with carbon number 5 of sugar through the oxygen atom. Okay, and this type of linkage, chemical bond, is known as the ester linkage or ester bond. So, this is how in a single nucleotide, individual chemical components are bonded with each other to form a single nucleotide. Next, we have to know how many nucleotides are actually combining to form a chain of nucleotides or polynucleotides or nucleic acids, functional nucleic acids. Now, see very carefully that phosphate groups, which is attached with the carbon number 5, which is attached with the carbon number 5, follow the cursor. Now, the phosphate group in a single nucleotide, phosphate group is attached to the sugar through the carbon number 5. That phosphate group is again forming another bond with the carbon number 3 of the sugar of another nucleotide above of it. So this pattern is repeated several times, again and again. In this way, significantly long chain of nucleotides is forming, which have a sugar phosphate backbone. And the center, in center it projects all the nitrogenous bases, like an array. Now, if the sugar deoxyribose, it is DNA. That means the carbon number 2 of the sugar is containing one hydrogen bond instead of hydroxyl group. And if it is a ribose sugar, then it is known as RNA. Now, all of you know that DNA is a double-stranded polynucleotide molecule. That means DNA composed of 2 polynucleotide chain, where primary structure of RNA is always composed of one single polynucleotide. Now, how 2 polynucleotide chain are bonding with each other? The answer is that the nitrogenous bases of a polynucleotide chain form hydrogen bonds with the complementary base of another polynucleotide chain. In this way, 2 polynucleotide chain chemically combine with each other and form double helix structure of a single DNA molecule. Now, what is complementary base pairing says? So, the complementary base pairing says that the nitrogenous base can only form hydrogen bond with their complementary bases. It is extremely specific. Here you can see that the adenine form hydrogen bonds with the thymine or uracil, where guanine can form hydrogen bond with only cytosine. So this session is up to this. These are the references. Thanks to all of you.